Good afternoon, everybody. I'm Adrian McConnell. I'm the CEO of the CO Research Trust. We're here at the final lecture of this year's series, and um, this year, oh, sorry, this time, I'm really pleased to be joined by Professor Derek Steele from the University of Leeds. Derek's been undertaking work funded by the Sierra Research Trust, looking at mechanisms within the heart, looking at the electrophysiology. Derek's background is looking at the molecular and cellular mechanisms governing excitation, contraction, coupling in cardiac and skeletal muscle. And also he's been looking at how these processes are modified by physiological changes or disease. Um, so Derek's here to talk about the work that he's been undertaking, and he'll be joined for the question and answer by his colleague, Dr. Moza Al-Awais. Um, a little bit of housekeeping. If you have any questions for Derek on the presentation, if you could pop them in the chat at the bottom, and what we'll do is we'll address those all at the end. Anyway, I think that's enough from me, and I'm going to hand over to Dr. Steele now. Thank you. Hi, Derek. Thank you. The floor's yours.
Excellent. Thank you, Adrian. I'm just going to start up the presentation. There we go. Okay, thank you all for coming today. What I'm going to be doing is I'm going to run through some of the work we've been doing funded by CORT on the carbon monoxide, on the effects of carbon monoxide on the heart. What I'm attempting to do, rather than just throw a whole load of our recent data at you, we've selected particularly interesting parts. And I'm going to give you a bit of a little bit of background to how we started this work, and then I'm going to give you a little bit at the end where I try to put the findings from this work into context and explain why I think it's important and where we might go from here. So it has been reported on numerous occasions, particularly from clinical case reports, that patients admitted with acute
Sorry,
cardiac.
Derek. Derek, so
Yep.
I'm sorry. Can I interrupt a second? We seem to have sort of grey boxes on your slide deck. I don't know whether it's something to do with the Zoom
Ooh.
presentation thing. So would you mind unsharing and resharing and see if that gets rid
Yep, let's try that.
of sorry about
Okay. We did try this earlier and it
that.
seemed to
Okay.
be okay.
And Carlos, who's one of the attendees, he can see it clearly on his side, so it might just be a us problem.
Um,
Yeah, we've still got them here. But if everyone else cannot see isn't seeing them and you
so I
don't
now
see them,
can't see. Okay, yeah.
okay,
So,
let's keep going. Sorry.
okay, sorry about that if it's obscuring the thing. We didn't see them earlier on. Okay, so in case reports, there are numerous case reports out there of people who've been exposed acutely to carbon monoxide as a result of poisoning. They quite often experience cardiac rhythm disturbances, in other words, cardiac arrhythmias, and a number of different types of arrhythmias that have been identified. Sinus tachycardia is when the heart is beating via its normal rhythm, where the rhythm is initiated by electrical activity in the sinoatrial node in the right atrium. They have also seen atrial fibrillation, where the atria at the top of the heart start to behave, basically beat out of sync. And most seriously, they have identified ventricular arrhythmias, premature beats, ventricular tachycardia, and in some cases, ventricular fibrillation, which can indeed be fatal. So this is an example of a type of clinical reports you see out there. Typically, what's been reported is prolongation and increased dispersion of the ECG, the electrocardiogram QT interval. you get an increased heterogeneity of ventricular repolarization. This is often a precursor to ventricular arrhythmias. So just to explain, what I'm going to be talking about today is going to be all about cellular recordings. And I just want to give you some idea about how the cellular recordings relate to the electrical activity in the whole human heart. Now, this shows the type of slide that I use to teach undergraduate students about the contraction of the heart and how it's initiated. And what this actually shows in yellow is how the heartbeat is initiated by spontaneously active cells in the sinoatrial node up here in the top right hand side or top right atrium here. And what happens during each beat of the heart is that this wave of electrical activity known as an action potential spreads first of all across the atria and then down via the interventricular septum via the left and right conducting bundles, and then up via the ventricles. And the ventricles are the thick-walled muscular part of the heart, the most muscular part of the heart, which then contract and expel blood out into the pulmonary and systemic circulations. What this diagram is actually doing is it's showing you at each time point here how this relates to the ECG, the electrocardiogram, which is, of course, used clinically to study how the heart is behaving and used, obviously, to diagnose pathology. And this is the configuration of the ECG that you most commonly see, the lead 2 ECG. Now, you have various parts of the ECG which are easily identifiable. The first wave, the first upward bump here, is the P wave, and that is associated with atrial depolarization. You then have the QRS complex, which is associated with ventricular depolarization, and finally the T wave over here, which is associated with repolarization of the ventricle when the electrical activity goes back to the starting point. So you can see there is a very strict correlation between where the electrical activity is happening in the heart and the recorded ECG. And trained clinicians can use changes in the ECG to understand, if the heart's behaving normally, if there are abnormalities in the heart. And indeed, clinically, there are many, many leads attached to the heart, if you like, viewing the heart from different angles. And you can use that information to identify, for example, if there was a particularly abnormal, if there was a series of abnormal events happening somewhere in the ventricle, you can actually use it to locate where the abnormality is happening. For this particular talk, I just want to really highlight here the relationship between the QT interval and why this is important and how it changes during disease and also exposure to drugs or toxic compounds that induce arrhythmias. So the QT interval can become prolonged. So this is the time between the beginning of this wave until the end of ventricular repolarization. And QT prolongation is a very common fingerprint of something that's happening to the heart that is likely to cause arrhythmias. So in this particular case, this is from a paper way back in 2008 talking about QT prolongation. And it shows here that this gap between the beginning of ventricular depolarization and the end of ventricular repolarization can increase under various circumstances. And if you look at what's happening to the individual ventricular cells in the heart, you can actually see corresponding changes in the ventricular cells. So this is an action potential. And this is the electrical event that underlies the propagation of this activity through the heart, which precedes contraction. So you can actually record these from individual cardiac cells. And if you see, following a particular intervention, a pronounced prolongation of the action potential, this is likely to correlate with QT prolongation in the intact heart. And it's likely to be a predictor of ventricular arrhythmias. And as you'll be seeing shortly, you can also see other abnormalities in individual cells, which give you a clue about what might be happening in the heart and the underlying mechanisms. Normally, I would give the acknowledgements at the end, but I actually felt on this occasion it was appropriate to start by telling you a little bit about the history of carbon monoxide research at Leeds. This is my collaborator, Chris Pearce, who sadly passed away in 2008, and he was the person that really kicked off carbon monoxide research at Leeds over 20 years ago. And during the time between around about 2006 and 2018, he actually published over 30 papers and review articles in carbon monoxide. He had a very broad range of interests. He was interested in the effects of carbon monoxide on neuronal cells and smooth muscle cells and in cardiac muscle. And although I'm not going to go into any details here, his initial interests were actually studying endogenous, naturally occurring carbon monoxide production in the body. Carbon monoxide, we shouldn't forget, is actually a naturally occurring substance, and it is believed to have important regulatory properties inside the body. And that's what Chris started to study roughly about 30 years ago, around the time that I completely fortuitously moved into a laboratory right next to him. And one day Chris came through to speak to me because he had some early samples of a compound called CORM, which could release carbon monoxide. And he was interested in potential protective effects of CORM in other tissues. And he asked me to have a look at this CORM compound because we were using paste isolated rat cardiac myocytes. And to our surprise, we discovered that when we added this CORM compound, it actually induced arrhythmias. And that kick-started collaborations that lasted with Chris until 2018, and then work that we've carried on beyond that. As I say, Chris was interested in many different angles here. The cardiac studies have involved a lot of different people. I don't have time to go through all of them today. I just want to mention in particular Mark Dallas, who actually I think published the first paper with Chris around about 2008 in neuronal cells. And Jacobo Ellis, who's now over at Bradford, he carried out some of the key electrophysiology work, as did Moza Alois, who is here today and who is the research fellow funded by the Carbon Monoxide Research Trust to carry out the work that I'm going to be showing you today on cardiac stem cells, human cardiac stem cells. So it was Chris that kicked it all off, and it's resulted in a huge amount of collaboration and some very interesting findings. So I just want to show you a couple of slides from our early studies on animal work. And this kind of explains why we wanted to go and look at human stem cells. This shows recordings of the action potential in isolated rat cardiac myocytes. And it shows an experiment. The control response is shown here in black. So this is the action potential in this cardiac myocyte. It's been repeatedly paced. And you can see that when we add carbon monoxide, descending phase of the action potential is quite markedly prolonged. And as I say, this is a signature sort of worrying effect with regard to the potential to produce arrhythmias. And indeed, we do see a particular type of arrhythmia here in the rat cells, where the cells, rather than repolarizing normally, in some cells, they exhibit this oscillation known as an early after depolarization in the presence of carbon monoxide. Excuse me. This shows the prolongation of the action potential, cumulative data. And when you see APD, that's action potential duration, at various time points during this descending phase, so either at 25%, 50 or 90% of the descending phase back to zero again. And you can see there's a very marked prolongation here. What we discovered was that these effects in rat cells were primarily caused by prolongation of the sodium current. So these are sodium current measurements. And the initiation or activation of the sodium channels happens very early on. It is the first event that really initiates the rising phase of the action potential and its propagation through the heart. This black line here shows a control recording of the sodium current in these rat myocytes. And in the presence of CORM2, you can see that it's smaller and it's also very prolonged. And in the case of the rat myocytes, it turned out that the decrease in amplitude was not particularly important, but the prolongation was. And it's this prolongation of the sodium current that was prolonging the action potential, leading to arrhythmias. We collaborated with a group over in France who did experiments showing that the ECG, you had QT prolongation in the ECG and arrhythmias, and that you could actually block this effect using a drug, an existing drug known as ranolazine, which was actually designed to inhibit the sodium current, but a particular part of the sodium current is late activating prolonged phase, and it could actually switch off these arrhythmias. So this was really quite fascinating to us because it gave us some clues regarding the what the underlying mechanism might be, but also introduced the idea that it's possibility that existing drugs out there might be useful in terms of treating acute exposure to carbon monoxide. So just to again focus here on this, two effects in rat, a reduction in the peak sodium current and this prolongation, which serve to prolong the action potential and underlies the arrhythmias, and it can be completely reversed by ranolazine. So we got very excited about this and published it. The next thing we did, however, was we went on to look at effects in the guinea pig. Now, I'm going to talk about this in a little bit more in a minute, but one of the reasons we wanted to look at guinea pig is because we knew that rat myocytes had some very significant differences to human myocytes. And in particular, one of the things that rat myocytes doesn't have very much of is a potassium channel known as hERG. Some of you might well have heard about hERG if you're vaguely involved in medicine in any way, because the hERG potassium channel is the nightmare for drugs companies because many drugs are ditched during development because they inhibit hERG. The potassium channel, this is a repolarizing current, and inhibition of this current destabilizes the action potential. It prolongs it and it destabilizes it, and it can lead to arrhythmias in humans. And basically, drugs companies very, standardly screen for hERG effects, and they're very reluctant really to proceed with any drugs that affect hERG, and that has resulted in quite a lot of drugs that could actually be useful being ditched if they have other effects which counter other beneficial effects which can counteract this. There's some good examples of this. But anyway, we saw a similar prolongation of the action potential in these guinea pig cells. But what we found was, importantly, we couldn't block this effect by using ranolazine, which acts to reduce this prolonging effect in the sodium current. And what we found was that that the carbon monoxide in this case was actually the dominant effect was inhibition of hERG. And you can see we got these EAD arrhythmias. So this is the control action potential here, prolongation. And in some cells, we got these EAD arrhythmias, which is a symptom of the fact that the cells can't repolarize. So this raised alarm bells with us because we had found, similar effects, arrhythmias in both rat and guinea pig. But as is so often the case, we remained uncertain about what the important effects would be in humans. So this is just basically saying that our past work in animal cells showed that carbon monoxide targets a number of things. It targets the sodium channel, it inhibits the peak sodium current and prolongs it. It seems to target the hERG potassium channel, and also we've seen effects on the L-type calcium channel, although broadly speaking, these effects are unlikely to be particularly problematic. If anything, they might actually be anti-arrhythmic. We have this problem though, because in animal models, differences in ion channel expression often make relevance to human heart uncertain. And this is exactly the same for us as it is for drugs companies over the years. The drugs companies very commonly use animal models in early stage trials to try and identify, for example, antiarrhythmic effects. And they are always left having to move closer and closer to actually giving the drug to a human to be certain that it's safe. So the early stage clinical trials, one of the reasons is to actually make sure that the results they have quite often in animal tissue do not cause problems in humans. So it's a constant problem, the difference between animal models and humans. So ideally, experiments would all be carried out on primary human cardiac cells. The problem is that primary human cardiac cells are not really available for routine research and certainly not healthy human tissue. This figure right here shows the action potential and where these currents are activated during the action potential to produce these changes. You can see that the sodium current is present here very early on, causing this rapid depolarization. You have the L-type calcium current, which occurs during the plateau phase, and you have IKR, the hERG potassium channel being activated to repolarize the heart, as shown here. So the aim of this project, funded by CORT, was really to start studying the effects of CO in human-induced pluripotent cardiac stem cells. These cells express human ion channels and the currents INA, IKR, and the calcium current, and regulatory agencies are increasingly using data from these cells to predict clinical proarrhythmic risk in man, and that includes the NIH in the States. And importantly, human-induced pluripotent cardiac stem cells have been shown to predict accurately the effects of numerous drugs and toxic agents in humans, including the actions mediated via hERG and the sodium channel. So this struck us as a very good idea to have a look at carbon monoxide and see what it does in stem cells. We are building upon years of investigations from an international consortium that started in 2013, the CEPA Comprehensive In Vitro Proarrhythmia Assay. And this was a series of a large group of research groups, international research groups, who set about trying to improve the situation and moving away from animal models which are inherently problematic. And there are three pillars to their approach. The first was to express ion channels in non-cardiac cells and to then measure the effects of drugs on these ion channels. The second approach was to take this information and incorporate it into in silico computational models of the heart to see whether or not these effects are likely to result in arrhythmias. But the third was that they were going to attempt to use human cardiac stem cells as a better model of the human heart to predict what the effects would be in humans. And in actual fact, they started this work going the other way around. So they started with a collection of drugs where they already knew, some of these drugs are 20, 30 years old. They already knew, they'd been through clinical trials. Some of them had problems getting through clinical trials. They knew some of these were high risk in humans. They knew exactly what they do in humans. So what they were doing was they were going back and pretending they didn't know this information, testing them out extensively in human cardiac stem cells and seeing whether the stem cell data would accurately predict the effects in humans, the known effects in humans. Now, this is the first list that actually went up to about 25 compounds, and one of the compounds they looked at actually was flecainide, which is a sodium channel inhibitor that I'm going to be talking about shortly. So our idea to work in human cardiac stem cells was really building on a huge initiative started by the NIH in the States. What we're doing here is patch clamp recording, or rather the postdoc in this, the research fellow in this project, Moza Aloes, was doing this, patch clamp recording. Carbon monoxide is released from this CORN2 molecule, although we have done confirmatory studies using carbon monoxide itself. we're looking at the effects on action potentials. We're also looking at effects on the ion channels that underlie the action potentials. This slide shows stem cells beating spontaneously. So these are cultured cells. You can see that anatomically, they don't really look like adult cardiac cells. And it actually surprised me when I started seeing this data coming out that there was such a good model of the effects of drugs on the human heart. But they are. And you can impale these cells and patch clamp them and measure action potentials. As you can see here, these are action potentials arising spontaneously in these stem cells. And you can put them in culture and make these recordings. What you can also do is pace the cells. So rather than allowing them to go themselves at this intrinsic frequency, you can actually pace the cells by injecting current and dictate the frequency of beating. And we've done experiments under both of these conditions. So these are recordings of action potentials in HIPSC, human-induced pluripotent cardiac stem cells, under control conditions. And then up here, if I can get my pointer, after 2 minutes exposure to this CORM2, carbon monoxide releasing compound at 10 micromolar. And these are in paste cells. So we're dictating when these depolarizations actually happen. And then this is the action potential that results from that. And you can see after a couple of minutes exposure to CORM, not an awful lot has happened. However, after 6 minutes exposure, there are very profound changes. So there's actually a rise in the baseline here, which indicates we suspect that there is a bit of tonic depolarization going on here. We haven't investigated this in great detail, but that's something I can maybe talk about later as another possibility. But what you can see here is the action potential amplitude has gone down markedly. And this is a very big effect, bigger than we saw in rat or guinea pig, which often the action potential amplitude wasn't particularly affected, but the duration of it was. You can also see there is a stimulus artifact right at the beginning where we are stimulating these cells. You maybe see a hint of it here and here. You can't really see it here because the rising phase of the action potential is so fast, you can't really see it on this scale. But in the presence of CORM, or CO, you can actually see that the stimulus artifact is here, and that's the rising phase due to the stimulus artifact. And the rising phase of the action potential is now so slow that it's separating from this. So it's slowing the rate of rise. It's also prolonging the action potential. This shows the probability density here. The black data are under control conditions, and this shows the action potential duration at 90% of the return to baseline. That's shown in red here, and you can show that what it's doing is it's prolonging the action potential, and it's also becoming more variable. And this is the APD increase at 0.5, half the way down here, and 90% of the way down here in the presence of CORM2. Roughly at halfway down, you're getting about 10% prolongation. At 90%, however, you're getting closer to 40%. So these are really quite big effects on the duration, but surprisingly large effects on the rate of rise of the action potential here. This shows the, sorry, let's just check. Yes, so this is now in spontaneously beating cells. So these are cells, oh, wait a minute, this is just, yeah. So this is paste cells again, control, and you can see the slowed rate of rise. And then here you can see this spontaneous event that has arisen here. Now, what you'd expect is that the event would actually happen here somewhat later than this, but the spontaneous event that's happened earlier is an indication that there's some form of effect which is destabilizing the membrane potential of the cell and leading to spontaneous activity. Another indication of prorhythmic activity. So this shows spontaneous activity and evoked action potentials here, and it shows that there's a very marked increase in this spontaneous activity in the presence of CORM2. So these are now spontaneously active cells. So these are cells beating under their own steam at their intrinsic pace. And you can see that under control conditions, the interval between these spontaneous action potentials is very regular. This shows after introduction of 10 micromolar CORM2, And you can see that there's a hint here that the regularity is becoming, well, it's becoming less regular in terms of the interval between these events. And then after further exposure to CORM2, what you can actually see here is that we're getting a reduction in the amplitude of the action potentials, but also these breaks in the trace here show periods that we have actually cut out because we wouldn't be able to get the trace in this piece of in this slide here because it basically became quiescent for 800 milliseconds in between here, or 2.6 seconds here. So the regularity of these spontaneous events, well, it became much less regular basically. And this shows an example in a cell measuring the APD at 90% reduction here. You can see under control conditions, the APD is very constant. And then after introduction of CORM, it progressively starts to increase. This shows the probability of density and of the inter-event interval. In other words, the time between these peaks. And you can see with the white distribution here, this is under control conditions. And after you introduce CORM, basically the interval between these events becomes very irregular, it becomes much more dispersed. You have early events and you also have delayed events in the presence of carbon monoxide. Again, indications of proarrhythmic activity. So we want to look now at what might be underlying these effects on the action potential, and bearing in mind that the action potential is the wave of electrical activity that passes through the heart to initiate contraction. We need to understand what underlies the proarrhythmic effects on the heart and the effects in the action potential, because ultimately what we would like to do is to try and work out if there is a way potentially of offsetting these effects, of treating these effects in patients who have been acutely exposed to high levels of carbon monoxide. So we have to start looking at the ion channel activity that underlies the action potential, looking at the effects of carbon monoxide to see what the dominant influence is in human cells. So this shows a protocol that Moser set up where we're using a ramp change in membrane potential using patch clamp. We're exposing it to a ramp depolarization from 100 millivolts up to plus 60. Now, when you do that, as happens during the depolarization of the action potential, you start to activate channels. Initially, you get activation of sodium channels, and then during the plateau phase, you get activation of the calcium channels. If you record the currents during this voltage step, you can actually see this. You can see the large early spike that's due to sodium channel activation, and you can see the secondary activation, which is predominantly due to activation of L-type calcium channels. The advantage of doing it this way is that you can very quickly see, you can do a ramp exposure like this. It only takes 500 milliseconds. And then you can repeat this ramp again and again and again. And you can look at the amplitude of these currents as a function of time during exposure to carbon monoxide. Now, an important aspect of this is you have to be sure you're actually looking at the right channel here. And one way of doing this is to use very specific blockers of the L-type calcium channel, nifedipine, or this blocker of the sodium channel, A803467. So we know from years of past experience that these are very specific blockers of the sodium and calcium channel. So you can do this protocol and you can check to make sure that these peaks are actually due to INA and ICA. And this shows data from a single cell where this ramp protocol has been repeated again and again and again, and the amplitude of these currents has been plotted along here. Now they're in slightly different scales for the sodium and calcium currents here, so that the control responses can be sort of lined up both in terms of the calcium and sodium channels. But what I hope you can see here is that when we introduce these inhibitors, Both the sodium current and the calcium current, which start around here. Now, remember, zero is up at the top here by convention, but you can see that these currents very rapidly go to zero after exposure to the inhibitors. So we can be sure that this is indeed the sodium current and this is indeed the calcium current, and we have a methodology that allows us to, look at the effects on these currents over time. So this shows in black here, now we have a recording of the sodium current under control conditions, and then after exposure to carbon monoxide. And you can see there's a very marked inhibition of the peak of the sodium current, not so much of an effect on the prolongation as we actually saw in rat. And this shows the calcium current component under control conditions and in the presence of carbon monoxide. And again, you get a very marked inhibition of the calcium current. And this shows the percentage change in the sodium current and the peak sodium current and the peak calcium current. So down by about roughly 47% in terms of sodium current. That is a very pronounced inhibition of the sodium current, and probably roughly about 35% in the calcium current. And this shows cumulative data again. So this is using that ramp protocol to record sodium, which is in black, and calcium in red under control conditions. And it shows that in the presence of CORM, it's basically a very marked progressive reduction in the amplitude of these currents as we move through time along here. So the other thing we want to look at is HERG. Remember I said in guinea pig, carbon monoxide inhibited the HERG potassium current, which is important in repolarization of the heart. In the interest of time, I'm not going to spend a huge amount of time explaining this, but basically, this is a standardized protocol that's used to initiate the HERG potassium channel current in myocytes. It involves going from -80 to more stepping up to more positive currents, usually up to a maximum of plus 40 millivolts, and you get this initial response. But in actual fact, the HERG component is revealed when you subsequently repolarize down here to -40 millivolts. And it's this part here, this bounce up that you get, is known as a tail current, which is actually the HERG potassium channel. So this is how the experiments are standardly done using patch clamps in all different cell types. Now what you can see here is we wanted to check that this is definitely HERG we're measuring here. So we've introduced a pharmacological inhibitor of HERG, and you can see that, well, this here is in the same cell following introduction of the inhibitor, and you can see that this peak here is markedly reduced in the presence of the inhibitor, and this is simply superimposing two responses at a step change to plus 20 millivolts, showing the inhibition caused by HERG in the tail current over here. So we have a method to identify HERG. What does carbon monoxide do? Well, the traces are slightly more noisy, but hopefully you can see the peak of IKR is here, the tail current is here. In the same cell, following addition of CORM, you have a marked reduction in the amplitude of the HERG current. So it's definitely inhibiting HERG as it does in the guinea pig. And this shows two selected superimposed responses from here at 10 millivolts, under control conditions, and then in the presence of carbon monoxide, showing this inhibition of HERG. And this shows the percentage, mean percentage change in the tail current at 10 millivolts, shown here. So quite a substantial reduction in the HERG current. Okay, so what I want to do for the remaining 5 minutes is just to spend a little bit of time telling you why I think these results are interesting, trying to put them in the context, explaining why they occur and what the relevance might be to humans, and also perhaps giving some clues about where we could go next in terms of firming up and a possible approach to dealing with arrhythmias in humans during acute toxic exposure to carbon monoxide. So I've got 3 summary slides. I know sometimes when I'm in talks, my heart sinks when someone has conclusions brackets 1, but hopefully these aren't going to be too arduous. So the dominant effects from my perspective looking at this, so in the human cardiac stem cells during repetitive triggered activity where we're dictating the pacing rate of the cells. Carbon monoxide slowed the rate of depolarization of the action potential and prolonged the action potential. It also markedly decreased the amplitude and it induced spontaneous events, which is a type of proarrhythmic activity. In spontaneously beating HIPSC cardiac myocytes, carbon monoxide markedly slowed again the upstroke of the action potential and it induced chaotic activity. The interval between the beats became much more variable. And again, this is an indication of a proarrhythmic effect. In terms of the effect on the underlying currents, carbon monoxide had major inhibitory effects on currents carried by key sarcolemmal ion channels that we've looked at, INA, ICA, and IKR, the hERG potassium channel, which influences shape, duration, and frequency of the action potential. So what is the relevance to the human heart? Well, the inhibitory effects on the sodium channel and IKR, the potassium channel, are most likely to underlie the proarrhythmic effects in humans. And these are the effects that we see in these HIPSC cells. Compared to effects we have seen in rat or guinea pig, the inhibitory effect of carbon monoxide on INA is much more pronounced. So it's a bigger effect in INA. We didn't really see a slowing of the rising phase of action potentials in guinea pig or rat. And there's also this, there's a very pronounced reduction in the amplitude of the action potential, and this tended to happen. I haven't gone into the concentration dependency, but it tended to happen in these human cells at lower concentrations of carbon monoxide than were needed to induce effects, certainly in the rat and to some extent in the guinea pig. So it's more sensitive to carbon monoxide. There are very big effects on the sodium current. If we assume these large effects on INA seen in these human cardiac stem cells translate to human ventricular cells, this would be expected to be proarrhythmic. So why do we think it would be proarrhythmic? So at this point, it's quite useful to look at what we know about the effects of other, for example, sodium channel inhibitors or conditions where you've reduced sodium current in the heart. So in the human heart, the sodium channel inhibitor flecainide, a drug developed in the 80s, is known to induce arrhythmias, particularly in susceptible patients. There's a very famous study called the CAST study, where they basically gave patients a flecainide and discovered they had to stop the trial because they discovered that a subpopulation of susceptible parents were actually dying as a consequence of ventricular arrhythmias. So we know that sodium channel inhibition, if it's big enough, can be extremely proarrhythmic, and that actually relates to slowing of the action potential as it passes through the heart and reactivation in a chaotic way of electrical activity when it should not be happening. Interestingly, there's an inherited condition called Brugada syndrome, where mutations in the sodium channel also lead to reduced sodium current and consequent arrhythmias. And Brugada syndrome can be treated using a drug called quinidine, which blocks a particular potassium channel, which counteracts to some extent the effects of reduced sodium current. And this might be a direction to look. So what I would suggest on this is what I would be interested in seeing done in the future is it would be valuable to use, first of all, computational modeling to further translate the effects that we've seen in these human-induced pluripotent cardiac stem cells to the human heart. These are a very good model, we know, for drug effects in the heart, but it's not 100% identical to the adult cardiac myocytes. You can bridge that gap using computational modeling, which is now very sophisticated. So we'd like to plug these data into a computer model and investigate whether the effects of carbon monoxide might be treated, for example, with a drug like quinidine. And I would just finally like to thank the Carbon Monoxide Research Trust for funding this work. It's been a very enjoyable and very interesting project, and it wouldn't have happened without this funding from the Carbon Monoxide Research Trust. And I'm going to leave it there. I think we're pretty much bang on time. Okay, if anyone has any questions.
That's brilliant. Thank you, Derek. It's a real privilege of my job, really, to be able to, participate in these kinds of discussions and really test my GCSE in chemistry and biology. I always learn something and have something to go away and find out about. But thank you. That was really, really, really interesting. Some great work that you've been doing on this, and obviously it's the latest in a very, very long line or thread of research. We've had a question in the chat from Dr. Liam Ridge at Manchester Metropolitan University, who coincidentally I'm actually up in Manchester as I speak, and I met with him yesterday, and his colleague Dr. Fred Jones, who I believe worked at Leeds with you guys, and he's doing stuff to do with electrophysiology here in Manchester. I don't know whether I have the skills to be able to bring Liam in here, or whether Lauren can do it for me. But let me see. Promote to panelist. Here we go. Just going to bring Liam in so he can ask the question himself. He's a developmental biologist specialising in sort of cardiology, so he's much better qualified and placed to ask the question that he put in the chat. And I bet he's delighted that I've put him on the spot and brought him into this conversation.
Thanks, Adrian.
Thank you.
Thanks very much for that, Derek. Really interesting. I just had a question on the use of CORMs, really, and how much you know about, how much the molarity of CO which is being released by those molecules, and how it correlates with kind of gaseous exposure in parts per million.
Yes. Now, not surprisingly, this is a question we've also been asked by reviewers. And there is detailed information about how we worked this out in our 2012 paper on rat. The first thing I would say is there is also information out there suggesting that CORM might have other effects that are independent from CO. And we have done a load of experiments using solutions bubbled with carbon monoxide to check that the effects that we're seeing are definitely due to carbon monoxide. So that's the first thing. It's not a sort of chemical artifact, it's definitely carbon monoxide. In the 2012 paper, we describe the procedure for working out how much CO is in solution with CORM and also with the carbon monoxide bubbled solutions. Now, with the early rat experiments, the level of CORM we were using was equivalent to quite a high toxic level of carbon monoxide. So we're talking about between perhaps 350 and 500 parts per million, which would be sufficient to kill you within perhaps an hour or two exposure, but not immediately. The interesting thing about the experiments on stem cells is a lot of these effects, we've actually seen it down to, instead of perhaps 30 micromolar CORM, all the way down to 3 micromolar CORM. So we're looking at substantially lower levels. We need to go back and work out exactly what that corresponds to. We've used 3 and 10 in this study, but we're probably down at around 200 to 300 parts per million, I should say, in terms of the equivalence. Yep.
Thanks very much. Adrian, am I right just to ask one more follow-up question to that?
Of course, yeah, please do.
Thank you. Yeah, I was just really interested to know as well about if you've managed to investigate kind of how long these effects are kind of experienced by the cardiomyocytes. Is it kind of a legacy impact of that initial exposure, which then causes these arrhythmic responses further down the line?
We haven't done an awful lot of experiments reversing it. Moza, do you want to talk about this? Have you done any experiments where you've attempted to reverse the effects?
I have tried to wash it off. Can you hear me?
Yep.
Yeah.
Yes. So in this experiment, I've tried to wash it off just with the normal physiological saline, and unfortunately, the effects are not reversible.
Yep.
And a length of time, for experimental length time, by 10 minutes. All action, there is no spontaneous action potential, for example, can be seen from applying the CORM. So basically the cell is alive, but it can't beat anymore.
Yeah.
So washing it for up to 10 minutes after that did not recover any of the action, any of the spontaneous action potential. Yeah.
Yep, and that effect, that gradual movement towards quiescence
Quite, yeah.
reflects the inhibition of the sodium current. The cell
Yeah,
can't depolarize. And this is actually what really strikes me about this. We saw sodium channel effects in the rat and the guinea pig, but the effects in the human are very large. Now, what this suggests is, I think, the levels that we've used, we see effects after perhaps 5 or 6 minutes. And then if you leave it longer, they get progressively worse and they don't rapidly reverse.
Yeah.
What is probably also true is that you could go lower than that and longer exposures, and you would start to see equivalent effects at lower levels, but longer exposures. So, and as I say, what really strikes me about this is that the sodium channel effects appear to be most dominant in the human cells.
That's a really cool experiment to try. Thank you very much.
Thanks.
Thanks, Liam. I've invited Izzy Myers to join us in the room here. Izzy is chair of COMED, which is the medical and healthcare subgroup of the all-party parliamentary carbon monoxide group. Izzy's, as I think most people on the call will know who Izzy is and what she does, and she's been involved in CO research and policy since CO was invented almost. Which she will thank me for. But Izzy has kindly said that she'd like to ask a question. So Izzy, the floor is yours.
Thank you. Thank you, Adrian. And Derek, it's really lovely to see how what started off as a brief email has developed into something that's really really important, as well as really carrying on sort of the legacy, if you like, of where the department started. So to the whole team, thank you. It's really a pleasure and an honour for you to have done this. I was going to take the sort of my question a bit more into that sort of public health realm in terms of sort of what we see, and just some thoughts. And it's going to be more of a a discursive answer rather than a scientific definitive answer. But the work you've been doing has been in essentially adult cardiac cells from those that are not already compromised in any way. So we're looking at healthy cells. So this gives rise, obviously, to that sort of question of, well, what might we see in compromised cells and adult cells, but then also that element of what we might see in terms of developmentally. So bringing in the work that's going on in Sheffield, which you have some overlap with on the developmental side. Just thinking that if in hearts of babies, for want of a better word, where we don't see any structural damage, what we are going to perhaps see, I'm thinking, is a latent sort of vulnerability potentially down the road from the work that we understand from that we understand from the work that you've conducted. So it could be that there is damage, but it's not a structural damage.
It's possible. So what I would say is, again, going back to what we know about the effects of drugs that inhibit sodium current. We know that, well, the CAST study that I mentioned in the 1980s, where they looked at flecainide, a sodium current inhibitor, and were horrified to discover it was actually killing people. The people that were dying were people that had underlying ischemic heart disease. So the thing about ischemic heart disease is you end up with part of your heart that's underperfused. And in that region, as the action potential passes through that damaged region, the action potential slows. And if the action potential slows, there is a property of nerve cells and cardiac tissue that prevents the action potential going backwards. It's called refractoriness. So in other words, when the action potential is passing through the heart, immediately behind the wavefront, it can't normally be reactivated. But if you slow down the propagation of the action potential, as occurs in ischemia, there is a danger that it can loop back round again and reactivate. And this causes a well-known type of arrhythmia called a reentrant arrhythmia. And the reason it was killing patients with ischemic heart disease was that flecainide was adding to this slowing of the action potential during the ischemic region. It's making it even worse. And it was basically causing reentrant arrhythmias in a very high percentage of the patients that were not recoverable. So, given the size of the sodium channel effects, I would say anyone that has a heart defect, be it genetic or perhaps ischemic heart disease, anyone with Brugada syndrome, be highly susceptible to carbon monoxide. Anybody who has ischemic heart disease, you would predict would be more susceptible. I would also predict there's going to be some considerable variation in susceptibility just due to national human variation, and also the fact that people with ischemic heart disease, they can have a tiny bit they don't even know about, or they can have a big chunk that's been damaged, that's remodeled, and you get very abnormal things going on there. So definitely. With regard to development, that's a very interesting question. I don't know an awful lot about the effects of carbon monoxide or potential effects in development, but what I do know about is experiments carried out by a French group where they did long-term exposure to carbon monoxide and looked at what was happening to the heart. And one of the things that you do see is fibrosis, cardiac fibrosis developing over time. And this can be an extremely subtle effect, and you would expect it, perhaps not necessarily to be particularly noticeable initially, and quite possibly cause, longer term effects down the line. Yep.
Great, thank
Brilliant,
you.
thank you. I think we opened a bit of a can of worms there in terms of those
Absolutely.
that have already have that sort of cardiac vulnerability, and
Yeah.
that is actually really interesting, particularly with all the sort of other work that's going on as well.
Yeah.
So no, thank you. That's you could have carried on for a long time.
Indeed.
Thank you.
Thanks.
Great, thanks, Izzy. Okay. Well, we're coming up to 1:30 now. I don't know if anybody has any other final thoughts or closing remarks or comments they'd like to make, perhaps Derek or Moser, Isabella or Liam.
Could I perhaps just say again a big thank you to the Carbon Monoxide Research Trust. I think it was actually during COVID that I got the initial email from the Carbon Monoxide Research Trust to engage and have a chat about this. And it's been a tremendously enjoyable journey. And I just want to compliment you on the way that you run the Carbon Monoxide Research Trust, because unlike normal academic engagement, it's been a pleasure to get to meet such a wide variety of people who are at the sharp end often of dealing with people who have been exposed to carbon monoxide, whether it's people in the fire service, people in nursing, people dealing with pregnant women who exposed to smoke. It made a change, and it was a very enjoyable, experience, because it's quite often, from our perspective, we quite often don't see the sharp end of the conditions that we are actually investigating. And I think that's a really important thing that you've done in terms of putting it together. And again, thank you very much for supporting us and Moser over the past 3 years in this study, which we've really enjoyed doing.
Yeah, no, well, thank you. I mean, that's something that we really try to do. And I think the team and I quite enjoy being described as abnormal. That's by design. Sorry, Moser.
I just would like to thank you all. That was a pleasure, 3 years work. And I'm hoping to carry on the same area and take it further. And hopefully the publication will show you that in the near future.
Well, I mean, I do hope that you stay in touch with us.
Absolutely.
And keep him, involved in the work that we're doing and others are doing, and keep us informed of what you're doing.
Absolutely.
So, yeah. So I guess in closing, thanks to you both for all the work that you've done over the last 3 years. It's been a real pleasure working with you guys, and I think we've learned so much. And I think, people who have been party to previous presentations and your contribution at events, we've all learned a hell of a lot. And Derek, I understand that you'll be retiring this year, so I just wanted to say good luck, and I hope you enjoy your retirement, and you'll be much missed in these parts. So thank you to everyone for attending. Thanks to the team for organizing the event. Thanks to Derek and Moser for the research and the presentation today. Thanks to Liam and Izzy for their questions. One little bit of news. The CO Research Trust, we've announced our annual conference for the year. and that's going to take place on the 12th of November this year. It's going to be a one-day conference, and it's going to be in central London. There are more details to follow. We don't have a page up on the website at the moment, but if you aren't signed up to our newsletter, you can sign up to the newsletter on the website, and that's where all the information comes out, and you'll be able to find out more about that as we develop it. So thank you, everybody. I'm going to leave it there now, I think. Hope everybody has a really nice afternoon, and I look forward to seeing you all soon. Thank you.